called structure-activity relationships. And this is a very nice way of analyzing the relationship between the chemical structure of a molecule and its biological activity. So you look at a compound, you look at how changing the compound changes the activity of the compound, so changes to the structure. SAR analysis enables us to figure out which chemical group in a scaffold is responsible for evoking a target biological effect in the organism. SAR also allows us to modify the scaffold to generate analogous structures that optimize the potency of a hit-to-lead pharmaceutical. So that is, okay, I have one lead compound. Now let's propose 10 other compounds that are similar. And these 10 other compounds might be easier to synthesize. They might have increased potency. And they might have increased purity. And in general, might result in an even improved lead compound. And this is all thanks to SAR, structure-activity relationships. And today we will do a concrete example because I don't want this lecture to just be theory. That would be very boring. I want you guys to see that at Portsmouth Uni, we can do this in practice, okay? My lab can do this in practice. Let's look at the Chembl database. Chembl stands for Chemistry at European Molecular Biology Lab. So this is an initiative from the European Molecular Biology Lab. The Chembl database allows you to scan for potential compounds to a particular target. And here's the website. I'll move the video. Here's the website so you can access it yourself. You can go on now if you want. Here's how the Chembl database looks like. As you can see here, it has a search bar for you to type in compounds. It has filtering that's very precise, so you can filter according to all kinds of things. You can see what's in the database. So you can see here, there's 2.4 million compounds right now. And those are actually divided. So you can see there's 1.9 million small molecules. Then there's 400,000 that are not small molecules. Those can be proteins, antibodies, etc. Okay, so it's a huge database. Okay, now let's introduce the case study that we will use today. I'll come back to this in a second. So the case study today is the drug on the market is called Taxol, and the effect of multi-drug resistance on the efficacy of Taxol. So Taxol, and the chemical structure is here, is a chemotherapy agent that is used to treat both early stage and metastatic breast cancer. So this sounds very noble and very useful. Yeah. We're not looking at some, elective thing. This is really for people who are very sick. But it can be used at the very early stages. Now, unfortunately for us, there's a challenge. Then the challenge is that Taxol is pumped out of the cell by efflux pumps, and this causes multi-drug resistance. So here's the efflux pump, and here's actually a structure captured with Taxol bound. And we can zoom in and look at this. And so if we really zoom in with molecular modeling, we can see Taxol in yellow and its surrounding binding site. And these are the amino acid residues of the efflux pump. This efflux pump has a name. It's called P-glycoprotein. And we will look at that in a second. So this is not great. Let's see if we could slow this efflux, slow it down, or even stop it. So if we stop it, we would increase the bioavailability of Taxol in the cell. And that would, increase the potency of Taxol. There would be more Taxol at the tumor site. And so more cancer cells would get targeted. Okay. So to do that, we need to stop the effluxing. And to stop the effluxing, you need what's called a small molecule inhibitor. Okay, so you want to inhibit the efflux process. So how do we do that? Let's use the Campbell database for that. Yeah, so the solution here is let's use Campbell database to locate an inhibitor for this efflux pump to stop multi-drug resistance. So here's our SAR example, PGP target hits. PGP is the name of that efflux pump. So we will go to the Campbell database and search P-glycoprotein. Okay, and if we do that, and we select the correct human P-glycoprotein, the database search returns a list of ligands that have potency in PGP. And we haven't looked at the ligands yet. And so let's first look at this page. So you can see here we have a certain number of records. We can increase how many records there are, but it's going to give us the ligands by potency, which is really nice. So number one is the most potent to our target, called P-glycoprotein 1 inhibitor. And it will tell us first the chemical structure of the ligand. It will tell us the name. The name of this ligand is tariquidar. It will tell us that it is a small molecule, and this means that it is not a peptide, it is not an antibody, okay? It's not a protein, it's a small molecule, which is a category in drug discovery. It will tell us even the phase, the clinical trial phase that this small molecule has arrived at. So it's gone very far in the process. And then there's some more information. And then it will start giving us number 2, number 3, number 4. These are similar ligands ranked by something called similarity scoring, and I will show that in a second. And well, here we have the names: ensequidar, sosuquidar, valspodar, pyrrhicodar. First of all, the names are really similar. So this tells you that you're not crazy. Campbell has returned something very useful. It has returned analogous molecules of tariquidar with a slight less potency to PGP. This means that tariquidar is the one that stops the pumping the most. Then you have ensequidar, then you have sosuquidar, then you have valspodar. Okay, let's look at this a little bit. So here's our Campbell database. Then we click on compound number 1. That is the number 1 ranked PGP target hit, and it's called tariquidar. Here's the chemical structure. Here's the name again, tariquidar. Here's something else. You have the molecular weight. You have other things like that. Then you have next to it the list of similar compounds. And here's now the similarity score that I was mentioning. So similarity score of 100% is for the same compound, and then it goes from 0 to 100. 90 means very close. This is very similar to target number 1. This is the one called sosuquidar. And then it continues, and sequidar, etc. And you can see the similarity is really high. So these are really similar compounds, and they have strong potency on PGP. And in total, there's 11 similar compounds in the Campbell database. Okay, let's look at a little bit more information on tariquidar. So tariquidar has nanomolar activity, meaning it has a KD of 5.1 nanomolar on PGP. So it is really a potent inhibitor of efflux. It's nearly sub-nanomolar. This is very good. Something that would be in the micromolar regime would not be very good, but this is nanomolar. It means you need a very small concentration of the inhibitor to stop the efflux. Okay. Tariquidar is also a 3rd-generation inhibitor due to its advanced scaffold design, thanks to combinatorial chemistry and high specificity, meaning it doesn't easily go and target other cells. Because you don't want that. If you're trying to block efflux, you don't want something that's also going to block your heart cells, cell receptors, and cause a stroke, for instance, right? So you want a high specificity. However, it is not known how toriquidar inhibits PGP, and so you might want to use molecular modeling to give an answer to this. Okay, the second database that we will use today is something called PubChem. So PubChem is the Public Library of Chemistry, is run by the National Library of Medicine of the United States. This is through the National Institutes of Health, and it is free to use, just like Chembl is free to use. And here's the website, here's the link, and you can go on that now. We will go on that website and we will look up toriquidar. And let's see what PubChem gives us. So, according to PubChem, toriquidar is an anthracylamide derivative with multi-drug resistance properties. Toriquidar non-competitively binds to the P-glycoprotein transporter, thereby inhibiting transmembrane transport of anti-cancer drugs. Inhibition of transmembrane transport may result in increased intracellular concentrations of an anti-cancer drug, thereby augmenting its cytotoxicity. So what this means is that if you add toriquidar to your therapeutic, it will stop the efflux process, and so your anti-cancer compound, for instance, Taxol, will pass to the tumor site more efficiently because it is not being pumped out all the time. And then by increasing the concentration of the anti-cancer drug, you increase the efficacy of the drug, and more cancer cells will be targeted. So here are the physicochemical parameters that toriquidar has, and this is all found through PubChem database, and so this is incredibly useful. Let's just read some of the parameters. It has a molecular weight of 646 grams per mole. So yeah. It has a molecular formula, C38H38N4O6. It has a CAS number, and this is for buying it. So when you want to order toriquidar for your experiments, this is the number you use. So again, this is where PubChem is really useful. It has a Chembl ID, so this also means you can go on PubChem, get the Chembl ID, and then go to Chembl and immediately find the drug that you're talking about. It has things like a topological polar surface area of 111 Angstrom square. It has Roman charge of 0. And there's more information. So I hope this explained a little bit about Chembl and the PubChem databases. These are open source databases.